phytoplasmas also, pneumatides we have to discuss here, and that is here the pathogen interaction we have to discuss in this one, and control of plant diseases. Now we have to control the plant diseases. We have to discuss in the black. So black four here, disease that is the bacteria, virus, and phytoplasma, and another one is spiroplasma. We are discussing black three, right? So coming to the here general account of the fungi. What is the fungi means here we can discuss. So the thallus organization of fungi means what is the fungi here? That is the fungi is the plural of word fungus, which is derived from the Latin word that is the fungus. So fungi are the allophilous and heterotrophic and eukaryotic thallophytes. Means here lack of the chlorophylls are there. So, according to Alexopoulos, the fungi include the nucleated spore-bearing achlorophyllous organisms that generally reproduce sexually, and those filamentous branched somatic structure are typically surrounded by the cell wall containing cellulose or either chitin. Here, the fungi is main, chitin is present, the cell wall is made up of chitin only. Okay, so that is so made up of the chitin only. Here, yes, here. So now, okay. So PPT is okay now. Now, okay, is anybody? Hello? PPT is clear?
Yes, sir. Clear, sir.
Okay, right. So, so here, that is the thallus organization we have to discuss here. The assimilative body or stroma of the fungus is thallus. So that is the comparatively simple growth forms lacking the differentiation into two stems, root and leaves. Here we cannot see this one. So the thallus is majority of fungi typically consists of thread-like microscopic filamentous structure, which is the differentiate the direction over the within the substratum utilized for food. So each of these filamentous is known as a hyphae, which is made up of thin and transparent to tubular wall filamented. That one is protoplasm is there, and they ensure that the sister nuclei arising from the conjunctive division do not remain the same daughter cells, but is instead the segregated in such a way that the eukaryotes are restored in the daughter cells. So here and so so the branch of botany that deals with the fungi is called mycology. and that who are to deal these fungi, those persons we call as a mycologist. Okay. So that is Micheli is known as the father of mycology, whereas the Butler refer to the father of Indian mycology. The Butler, E.J. Butler, which is the famous pathologist or mycologist, is known as the father of Indian mycologist. So fungi are non-green in color with the capacity to live in all kinds of environment. So the fungi can grow in anywhere they can grow, right? So, so what is the difference between the algae and fungi? This one is the main uh, concepts. So how we are to
1 minute,
different?
sir. Please, other candidates, please mute yourself. It is a lot of disturbance.
Yes.
Other candidates, please.
So all of you mute itself, yourself only. All of you
Dashrant,
mute.
Power Dashrant, sir. Shirisha. Shirisha, ma'am, please.
So all of you remaining people to mute. Right. So the main difference between the algae and fungi. What is the main difference? Is anybody?
Nutrition. Algae
Okay.
is autotrophic, fungi is
Yes.
heterotrophic.
Very nice. That is good. And another one is also cell wall composition is also different.
Yes.
So that algae, yeah. So cell wall, that algae is made up of the cellulose, but here in the fungi, that is made up of the chitin. Main portion of the cell wall here, chitin is present. So what are the we are to discuss between the algae and fungi? Here, the process of chlorophyll, which is some of them masked by the pigments such as brown, red algae, and green algae are there. But fungi here lack of the chlorophyll, right? So another one, the chlorophyll they are able to synthesize their own food and thus called as autotroph. But here the lack of the chlorophyll, that's why they are called as heterotroph or parasite or saprophytic conditions are right. So, lacking chlorophyll, the fungi is unable to synthesize their own organic food material. They get it is ready, means the source either by living as a parasite or saprophyte. For this region, the fungi is refers as heterotrophic for their nutrition character. Based on their nutrition, that's why we call as a heterotroph. So thallus composition also, that is the true parenchyma is present. That is here the fungal thallus is termed as the mycelium. They are formed the division and differentiate into cells but composed of the false tissues called as a pseudo parenchyma. So the unit of structure of the fungus, the thallus is not a cell. That one we call as a hyphae, right? So here in the algae we call as the thallus, but here in the fungi that one we call as the hyphae. That is the difference. So cell wall another character here the cell wall composed that is the cellulose, but here the cell wall and are either hyphae that is the made up of the chitin only, right? So another character reserve food material after the photosynthesis they are the In the algae, the reserve food material in the form of the starch. But here in the fungi, in the form of the glycogen, the reserve food that is accumulated in the form of the glycogen only, right?
అమ్మ వచ్చిద్దిలే మరి ఇప్పుడు రాడంట
Right. So here remaining people, please mute yourself.
Sirisha madam, please. Sir, excuse me, sir.
So, right. So in the algae members, the reserve food material, the starch only. But the fungi, the reserve food material is the form of the glycogen. So next one, that is the habitat, the light is present. Means algae can grow, which is need for the light, for the growth. Means for the doing the photosynthesis and drinking. So, but the fungi, they can grow in the dark or dim light environment only. Why here? There is no need for the light. Means lack of the chlorophyll is there. So that's why they are the not depending on the light only. They are they are exhibits as a saprophyte or either parasite. So they can grow in the water or damp soil, sometimes as epiphyte or either endophytes. But coming to the fungi, there are the different habitat is present. So parasite in the tissues, that's the plants and animals only. But coming to the reproduction in the algae, that is the reproduction, sexual reproduction increase in the complexity from simple to the higher forms. Means from algae chlorophycy to rhodophycy. That is the simple structure to complex reproductive structures are present. In the chlorophycy, the simple structures are present. But rhodophycy, pheophycy, that is the very complex structure of reproductions are there. Means very complexity structure is present in the algae members. There are here in the fungi, the gradually simplification and ultimate elimination of the sexual apertures from the lower to higher fungi. Means here that is the simplification is occur from lower to higher, right? So coming to the similarities, what are the similarities between the fungi and algae? This is some basic concepts to understand the what is the difference between the fungi and algae. So fungi and algae both are
Sir,
the
1 minute.
preferable.
Sir, please don't zoom, sir. If it is okay, normal size okay, sir. We can see.
Okay, right.
Don't
Okay,
zoom.
right.
We cannot see if you zoom.
Okay, all right. So that is the both are the preferred for the moist environment. The benefits of algae diverge from their symbiotic lichens relationship with the fungi. They are able to survive on the land in the moist environment. So what is the lichens? Is anybody? Lichens, you know?
Symbiotic association between algae and fungi.
Okay, nice. Symbiotic relationship is occurred between the algae and fungi. Which one we call as a phycobiont and mycobiont? Is anybody?
Algae is phycobiont.
Nice.
Fungi is mycobiont.
Okay, very good. So here, the phycobiont and mycobionts. So algae we call as a phycobiont, fungi we call as a mycobiont. Both are the associated. to form the lichens. These lichens are environment pollution indicators. Where the pollution is high index is there, that lichens cannot grow. Means algae they can give the means food material to the fungi. The fungi to give the shelter to the algae. Both are the symbiotical relation is occur. Mutualism is occur. Right? So next coming to the fungi and algae, both are the thallophyte. What are the thallophytes?
No true roots, stem leaves.
Okay, good. So that is there is no morphogenesis. Means root, stem, leaves, the flower, and they cannot see. Morphogenesis is not there. Parts are not present. Only the simple structure is there. Thallus, only cell wall. The parenchymatic tissue is there, and the central cytoplasm is there. Then nucleus is present. Simple structure is there. We cannot see root, stem, leaves. Organogenesis is not present. That is we are called as a thallophytes. So both are the cryptogams. Is anybody cryptogams means?
Hidden reproductive structures.
Very good. So here the thallophytes all are the cryptogams only. Means here flowering system is not there. That one we call as the cryptogams. Plants are phanerogams, cryptogams. Two classes are there, plants. So phanerogams means flowering plants. Cryptogams means non-flowering plants. That is, both are the algae and fungi. Both are the cryptogams only. And another one is the eukaryotic condition. Both algae and fungi, both are the eukaryotic condition, except to blue-green algae. These are the prokaryotic condition is present. So prokaryote means, is anybody? The nucleus.
Well defined nucleus is absent.
Ah, so here the nuclear envelope is absent. Only the DNA, they are formed as a thread-like structure. A specific nuclear envelope is there, not there, not well developed. That is the nucleus is present in the prokaryotes. Eukaryotes, here a prominent envelope is there, and well organelles, cell organelles also present. So another point, fungi and algae generally that is the organism possessing applied nuclei. That one that is the applied nuclei means the single form of chromosomes are there. Okay, and coming to the vascular tissue is totally absent in the fungi and algae. Vascular tissues means here the xylem and phloem, cambium is not present. Means connecting tissue is not there. Well developed vascular system is not there. Only the simple parenchyma tissue is present. So both algae and fungi possess cell wall made up of the polysaccharides. That is the and the symbiotic members present in the both groups. That is the animal fungi with roots. Like examples, lichens and some Frankias also. There are different they can grow. Anabina. They also Anabina is also symbiotic relations occur. And they are the both groups means algae and fungi. They are to reproduce asexually. And here sex organs are naked. in the both group. So no protection cover for the sex organs in both groups here. So coming to the general characters of fungi. What are the here? We can see the some characters. First one is the occurrence. So occur means the fungi they can grow anywhere. Means cosmopolitan characters they can show us. In anywhere means any environment condition the fungi they can grow in the air, in the water, in the soil, either within the plants or either within the animals. In anywhere they can grow. So, but they can grow in the warm and humid places. Hence, they keep food in the refrigerator to preserve the bacteria and fungal infections. We know that. So here and the thallus organization. So some of the unicellular forms, example yeast and syncytium, and another one is the multicellular forms are there. So the fungal body of thallus is called as the mycelium. So see here, this one is the entire the cell is a single cell unicellular that is this, and some are the filament like structures are present. Example, muca, rhizopus. So mycelium is the intervenes mass of thread-like hyphae. The hyphae may be septate or sometimes aseptate condition is there. So aseptate, which is to occur the xenocytic condition we can see. Means in the number of nucleus are present. Aseptate condition here without cross walls. Without cross walls are there, so that's why here xenocytic condition is there. Means the entire the filament or the thallus, which is look like a tubular like structure. So some fungi they are to found in the dimorphic that is a unicellular and mycelial forms that is the candida. So here sometimes some thallus may be unicellular or filamentous structures are present. What is the unicellular? Right? So unicellular thallus, that is the lower fungi. The thallus is more or less spherical, single-celled
Sir,
structures are
please
there.
don't zoom, sir. We cannot see if you zoom. Ah, okay, sir.
So
Continue, sir. Thank you.
wait. So here, so here that means the all the cell organelles are within the single cell only. That is a spherical structure. When the reproduction time, that is the such fungi we call as a holocarpic fungi. In the unicellular holocarpic forms, the mycelium is absent. Example, syncytium. Some of the holocarpic fungi yeast, they are produced bud cells. Means that the succession and these remain attached to one another. They are formed in the chain-like structures. So the chain of buds is referred as a pseudomycelium. So in the yeast, that is the asexual reproduction is occur budding nature. But some other cell they are to produced as a small bud or small spherical like outgrowth. So that the daughter bud they are to form as a new mycelium. So that is the they are called as a pseudomycelium. So another, the filamentous forms, filamentous fungi. So the most of the true fungi, the thallus is filamentous and composed of hyphae. So loosely aggregated hyphae called as means collectively form a network structure that is we call as a mycelium. Example, rhizopus, mucor. So means that hyphae or the filaments or tubular like structures to form as a network like structure. We know that one. Here in the bakery bread. So when we purchase the bakery bread that open in the direct exposed in the environment, or we have to keep a 2 or 3 drops of water. We leave it around 1 or 2 days. What happened here? So that the black color, some spots are appear. So that one is the mucor or sometimes rhizopus. Black moles, box spots will appear. So means here that is the a network like structure is there. That one we call as the mycelium. So each hyphae may be very sharp in size and branching of hyphae is dichotomous. So on the basis of presence and absence of septa, hyphae may be mycelial fungi or two types are the septate. That is the septate. Means septa means a cell will be divided into two. Okay, na? So here the filamentous forms two types of hyphae are there. Septate hyphae and aseptate hyphae. So what is the aseptate means that mycelium is containing the numerous nuclei. Already we know that one. That one we call as a means xenocytic condition. So numerous nucleus are formed on the cytoplasm. So without cross wall in the hyphae, divisions are not there. So without cross walls. So example zygomycetes, oomycetes. Such condition we know the xenocytic condition. Mucor, we can see the xenocyte. So in the formation of the septa, sometimes the septa is formed at the formation of reproduction time. That is the there are two organs means zoospores are produced in the sporangium. When the sporangium is formed, a pseudosepta is formed. Allomesis, example. Okay. So sporangium is formed. The sporangium is separated from the these mycelium. Septate hyphae. So hyphae are septate and hyphal segments. Many contains one or two more nuclei. Example: Ascomycotina, Basidiomycotina, and Deuteromycotina. So according to the septa here, there are the primary septa and advantageous septa are there. So primary septa are formed in the association with the mitotic or meiotic nuclear division, and they are separated to daughter nuclei. These types of septa found in the Ascomycotina, Basidiomycotina, and other asexual stages. So advantageous septa, which is the formed in the absence of mitosis and meiosis. This one is the local concentration cytoplasm. These are found in the lower groups like Mastigomycotina and Zygomycotina. So just wait within 1 minute again.
నీదమ్మా ఒక్క నిమిషం. ఏంటిది ఇది? ఏంటిది? నాకేమో విషయం ఏంటది? పెట్టు. అందరు తెలవండి. నిజంగా నాకు కూడా విసుకొస్తుంది.
Other candidates, please mute yourself. Don't unmute, please. It is creating a lot of disturbance.
So now different forms of mycelium. So here the different structure of myceliums are there. So different forms of mycelium referred as the various shapes and functions of mycelium, which are modified according to the circumstance. So based on there, here the plectenchyma and prosenchyma, pseudoparenchyma. These are the different sclerotia, rhizomorphs, rhizoids, apressoria, astoria, hyphal. These are the some forms are there. Different forms we can see here means based on their shapes, structure, and functions also. Sometimes functions also based on their functions and structures of the mycelium. Here different forms are there. So first one is the pectenchyme. So what is the pectenchyma? Here in the fungal mycelium, the hyphae are organized loosely or compactly woven to form a tissue called as the pectenchyma. Means the tissue means the fungal hyphae or fungal mycelium, they are loosely or compactly woven like sponge. That one we call as the pectenchyma. So again here, that is the plectenchyma, two types are there, prosenchyma, pseudoparenchyma. So prosenchyma means in this fungal tissue, they are loosely interwoven, laying more or less parallel to each other. So there are the parallel to each other, that is the prosenchyma. Pseudoparenchyma means in this fungal tissue, hyphae are compactly interwoven, looking like a parenchyma in cross section. That is pseudoparenchyma. Plectenchyma, here two types are there: prosenchyma, pseudoparenchyma. Prosenchyma, that is means parallel like structures are there. But here, that is the pseudoparenchyma, that is what here there are the like cross sections means interwoven looking like a parenchyma into cross sections are there. Coming to the sclerotia. So sclerotia, there is a Greek word here. These are the hard dormant bodies is consist of compact hyphae that is protected by external thickening hyphae. So external, see here, this one is the The diagram, you can see that prosenchyma and pseudoparenchyma. Lectenchyma here, two types are there. Means prosenchyma parallel like structures are there. Okay, so that is the prosenchyma, and pseudoparenchyma means some here what here interval looking like a parenchyma. Okay, this is there. And sclerotia. What here that is the sclerotia means here a protected by external thickening hyphae, each sclerosium germinates into a mycelium on the return of the favorable condition. Example penicillium. In the penicillium here we can see these sclerotia. Right? Another one is the third form is the rhizomorphs. Rhizomorphs means root-like compactly interwoven hyphae with a distinct growing tips, which is help to the absorption and penetrate on the substratum. Rhizomorphs, you see that the diagram, third diagram is the rhizomorph means they are to observe the nutritions and penetrate into the substratum. That is the uh, rhizomorphs. And another one is the rhizoids. Rhizoids also there. So rhizoids is example we can see in the rhizopus or mucor. So in the rhizoids, a rhizoid is short root-like filament branched off the thallus, generally formed in the tuft at the base of the thallus. So this also function anchoring and observe the food material. So rhizopus are either mucor, we can observe these rhizoids. See here, these are the rhizoids. So these rhizoids to function to fix into the substrate of the fungi. Right? So next one is the Aprosoria. So Aprosoria, there is the single form, Aprosorium is the single form, Aprosoria is the plural. So it is the terminal simple or lobed swollen mucilaginous structure of the infected hyphae which is adhesive to the surface of the host or the substratum. So see here that the diagram we can see that is the Aprosorium is there. So these are formed by some parasitic fungi such as the powder mildew and rust. So the rusted that beet rust, sorghum rust, these are the fungi we can see these Aprosorium. So means some lobed swollen structure is appear on the surface of host tissues. Uh, so next one is the Hastoria. So Astoria is also plural word and Astorium is the single word. So a astorium is an organ that is developed from a hyphae, usually the that is the performing the function of absorption of the food material. So this one is the obligative parasite character. They are very in shape, maybe. So, so different shapes are there, maybe knob-like structures and secret some specific engines and button-shaped and elongated finger-like structures are branched. So, what is the main Astoria function? To observe the food material from the Astoria. So, this is the Astoria. A anchor-like structure, finger-like structures are there. They are penetrate into the entire the tissues of host cells. So, this is the Astoria. Okay, they are observed, they are branched and finger or some anchor-like structure, lobe-like structures are there. Okay, so these Astoria also sometimes they are secretly digestive enzymes also. Means the digestive enzymes are means hydrolyze and carbohydrates, their proteins, they are digested in the host cells. So next one is hyphal traps. Means that is the predaceous sponges develop sticky hyphae or network hyphal, groups known as hyphal traps or snares are there. They are the like helps in the capturing the nematodes. And another one is the stomata. So stomata are compact somatic structure, much like is the mattress or fructification of general formed on the these fungi. So coming to the structure of the cell here, this one is the structure of the cell. We can see the cell wall is a eukaryotic condition. We can see eukaryotic condition means the cell wall is determine the characteristic shape of cell. Is protect the cell wall as osmotic lysis and also act as a binding site for some enzymes. So here the chemical composition of cell wall is not the same in all fungi. Chitin is the characteristic present the cell wall of the most of fungi. Right? So the chitin of fungal cell is not only strictly identical, some animal chitin and is been suggested for the fungal chitin. So cytoplasm is the hyphal is the tip containing the Golgi vesicle called as cytosomes, which is filled with the cell wall material. So nucleus and mitochondria are found to connect with the endoplasmic reticulum. Nucleus divides by the intracellular mitosis called as the karyosis, where the nuclear envelope is remain intact during the nuclear division and internal spindle develops and reserve food material is the glycogen and oil bodies are there. So here there are the different species and compositions are there. So next one is the nutrition, mode of the nutrition. So fungi lack of the chlorophyll, therefore they are cannot synthesize their own food depending on the where uh, how they are get the nutrition means here in the form the saprophyte parasite conditions we can see and sort of predaceous and symbiotic conditions also there. in the form of the mycorrhiza we can see here. So means what are the saprophytic fungi? So saprophytes means they are obtained food from the dead and decaying organic matter. So these they are the secret digesting enzymes to outside, which is the digest the substratum and then observe the nutrition. Example, mucor, agaricus, rhizopus. These are the saprophytic fungi. So here means with help of the Astoria only, these observe the food material. So coming to the parasites. So parasites means they obtain food from living organisms. They may be facultative or obligative facultative. So parasites means obligative parasite or facultative parasites are there. So here there are the different types are there, mucor and remaining Yushtilago, downy mildew, pythium and Puccinia. These are all of the coming to the endophytic parasite and ectophytic parasites also present. Here different conditions are there. Another one is the predaceous. So predaceous, some soil fungi developed a ring-like noses to trap the annelids and nematodes. That is the we call as the predaceous. So symbiotic condition. Next one is the symbiotic conditions are occur. So there are the mutualistic relationship with another organism by the both benefited. So here there is the lichens and mycorrhiza, both are the symbiotic relation is occur. So what is the lichens here? Symbiotic association between the fungi and algae. The fungal pattern is the member of Ascomyces or Basidiomycetes to provide water and nutrition. While the algal pattern is to green algae or cyanobacteria, they prepare food to provide these fungi. So that is the mycorrhizas means that is the mutualistic symbiotic association between the soil fungi and the roots of most plant species. So that is they are the where the deficiency in the soil phosphorus and nitrogen. So there are to fix these phosphorus and nitrogen into the atmosphere. So here also mycorrhizae, there are two types are there, ectomycorrhizae and endomycorrhizae also present. Um, so coming to the another character here,
Sir,
heterotalism and
can you show last slide once?
so here this one is the and
Sir, please don't
means
zoom.
okay. So here that is the means the lichens, different lichens are there, and here there are the ectomycorrhizae and endomycorrhizae. Here we can see diagram. So now coming to the another aspect is the heterotalism and homotalism. What are the heterotalism means that black spee. These black spee is discovered main these two types of the strains in the mucor. Heterotalism mucors also there, homotalism mucors also there. So this Blacksky is the mycologist. He is find the different mating types called as the heterotally. So the fungi without mating types called as the homothallic fungi. Another strain will come for the reproduction process here. That is the heterothallic condition is there. So nowadays we call some fungi, algae, and homothallic if both and male and female gametes produced in the same individual can fertilize each other that and heterothallic if the gametes can only be fertilized by gametes to form another individuals of the same species. So coming to the reproduction here, so the fungi is the reproduction takes place by the three methods are there: vegetative, asexual, and sexual reproduction. Three methods are there. So during the asexual and sexual reproduction process, spores are there are essential structure. The spores are liberated from the sporangia, and the spores are formed after meiosis. They are called as the meiospores. Means the spore mother cell there undergoes to the reduction division to produce the four applied meiospores. So example basidiospores, sporangiospores, ascospores, and these are the meiospores. So those are resulting the mitosis. They are all called as the mitospores. Means zoospores, aplonospores, conidias, and another Fuchsinia urodospores. All are called here mitospores. Means there is no reduction division. And diploid body produced as a result of the sexual fusion of gametes. That one we call as zygote is formed, which is the lower fungi term as resulting the spore, oospore and zygospore. So in the higher fungi, zygote is produced is represented by diploid nuclei produced
Sir,
in
go to
the
next slide, sir.
so right here that is the different mm, reproductions are there.
Pinter, please stop annotating. Quant, don't annotate.
So coming to the here vegetative reproduction here vegetative reproduction asexual and sexual reproduction. Three methods are there in the fungi. So vegetative reproduction means here only the mycelium is separated and form the new individual hyphae. So from that vegetative reproduction is there. So again reproduction, vegetative reproduction again different methods are there. Fragmentation, fission, budding, and sclerotia, rhizomorphs. These are the different methods which is reproduced by the vegetative reproduction. So fragmentation means the hyphae break into small fragments. That the mm, fragmentation is there. So each piece or small piece that is suitable condition, it will get the new mycelium. That is the fragmentation method. Means the mother hyphae or mother thallus will be break into small fragments. That method we call as a fragmentation. Next one is the fission. So this one is the splitting of cells into two daughter cells. So the two daughter cells, they are to develop as individually and they are construction by the cell wall formation only. That is the fission. Means mother cell divided into two ops, individual ops are there. So next method is the budding method. So budding method we can see in the yeast cells only. So a small bud formed from the parent cells, which is gradually increased in size and receive the part of the some nucleus and cytoplasm from the mother cell. So a cell wall is formed, which is a separate in the daughter cells from the parent cells. Example, this budding we can see in the uh, yeast cells only. So next one is the sclerotia. So these sclerotia is
Sir, go to next
penetrate.
slide. Slide not changed. Okay,
So
sir. Thank you.
the sclerotia means these are the perinating bodies formed by the compact mass of the interwoven hyphae. So this is the suitable condition germinate to the new individuals. So example classes purpurea and sclerotia. So these are the developed individually. So another one rhizomops, another method in the vegetative reproduction. So root-like elongated mycelia strands, they are the in the unfavorable condition. When the favorable condition, they are developed into new mycelium. So these rhizomops also to produce the new mycelium when the favorable conditions is occur. That is the rhizomops. And coming to the what here, asexual reproduction. In the asexual reproduction here, there are the spores are either motile or non-motile spores are formed. Here, various spores are also formed in the asexual reproduction. Zoospores, sapronospores, sporangiospores. Already we have to discuss that. So zoospores means there are commonly found in the lower fungi. So they are nacre spores which is after warming or insist secrete a cell wall and germinate by the germ tube into the thallus. They are the liquid with the two flagella, some are the one flagella is there. The flagella may be pantonymatic or either acronematic flagella. Okay. So means tinsel or either that name also called as the tinsel and whiplash flagellas also there. So means tinsel flagella means that the surface of the flagella is very smooth. That is tinsel flagella. Whiplash flagella means the surface they have some mastogonyms, feather-like structures are there. This one is there. See, so whiplash and tinsel flagellas are sometimes two flagellas are either there. So next one is the sporangiospores. So sporangiospores are aplonospores. There are the non-motile and the lack of the flagella. There are formed inside the sporangium. Example, Mycorrhizopus, right? These spores may be uninucleate or sometimes multinucleate and possess two-layered cell walls. Aplonospores and sporangiospores. Both are the non-motile or non-flagellate spores. They are produced within the sporangium. And another one is the conidia. Conidias. So they are the produced extremely are branched, unbranched hyphal tips termed as the conidiophores. The conidia may be formed singly in the chains. The conidia chains may be basipetal or acropetal, basipetal or sorry, basipetal or acropetal manner. Means basipetal manner means from base to edges. Acropetal means from edge to base. That is the development of the basipetal and acropetal. So the conidia may be uninucleate or multinucleate. The latter they are more common in the members of the farm class Deuteromycetes. So next one is ideas also there. So they are produced with the fragmentation of the hyphae from apex to base. So see here the diagram here idea some tip to base here that one is the ideas are there. So each cell does form the round off and separate as a spore which is under the favorable circumstance. They germinate and form the new mycelium. So next one is the chlamydospores also there. This one is the chlamydospores. They are look like a chlamydomonas. They are formed rounding off enlarged termination of intercalary cells of the hyphae. They may be singular or formed in chain-like manner. They do not separate from the hyphal, but they remain the viable and germinate under the favorable conditions. Just like chlamydomonas also, like this condition is there. So that's why we call it the chlamydospores. And another one is the ascospores. So ascomycetes, we can see these ascospores, and ascospores are produced inside the ascus. So during the optimal condition, this kind of spores is a specific to fungi is classified as ascomycetes. So another one is the iridospores, as involved red and summer spores of rust fungus, means wheat rust. So there is we can see in these some grasses also, iridospores also there. And another one is the basidiospores also. So basidiospores, these spores are produced by the Basidiomycetes fungi. They are typically each containing one haploid nucleus that is the produced of meiosis. So Basidiospores also there. So coming to the sexual reproduction here, mm, it is the formation of fusion of two gametes. So the sexual reproduction found in all groups except Deuteromycotina. So Deuteromycotina we call as the imperfect fungi. Means here sexual reproduction is absent. So here these sexual reproduction here three distinct phases are there. Three stages are there. That is the plasmogamy, karyogamy, and meiosis. Plasmogamy, karyogamy, and another one is the meiosis. Three stages are there, or either three phases. So what is the plasmogamy? Means the fusion of protoplasm. Plasmogamy. Fusion of protoplasm. Plasmogamy. Next one is the karyogamy. So fusion of the nuclei, that is the karyogamy, means nucleus will divide into two, karyogamy. So another one is the meiosis, means the reduction division of zygote that produced the four haploid spores. So these three methods means three stages are occurred in the sexual reproduction of the entire fungi except Deuteromycotina. Right? So here we can see the some methods are there. Different methods also there. We can see in the reproduction, sexual reproduction. Here that is the planogametic copulation. Planogametic copulation. What is the planogametic copulation? Means this one is the simplest type of sexual reproduction. The fusion of two gametes, opposite sex strains, they are to place both fusing the gametes are motile as a result to form the diploid zygote. That is the planogametic copulation. So isogamous, heterogamy, and here also there means both gametes. Isogametes means both gametes are similarly in the same, means morphologically and physiologically is also same gametes are there. That one we call as isogamous. Heterogamous is anybody? Heterogamous means hetero means
Different. Different.
different means morphologically and physiologically both are the different gametes are there, right? So here the heterogamous means when the
Sir,
fusion
go
of
change the
gametes.
slide, sir.
So Morphologically and in the physiologically, both the different gametes are there. That one we call as a heterogamous or heterogamy. In that heterogamous, anisogamous and oogamous also there. Anisogamous means anybody? Aniso. Already we have discussed
Not
iso.
similar. Not similar.
Yeah, yes, good. So aniso means that is the morphologically similar, but the physiologically is the dissimilar. That is the anisogamous. One is very active, one are the very less active. Some difference is there, anisogamous. Physiologically, the difference is there. One is very, they can swim in very slow. One is very fast. And oogamous type of reproduction. So another one is the oogamous type of reproduction. One is the motile gamete is there. That is the anthrozoids or male gamete. Very small in size. And one is the large non-motile. That one we call as a female gamete or egg. So oogamous type of reproduction. Isogamous, anisogamous, and oogamous type of reproduction is occur. Right? So next one is the gametangial copulation or gametangial contact. So gametangial contact means in the gametes means gametangia. So gametangia opposite chain of sex come to contact with each other. So male gametangium transfer the male nuclei and the female gametangium is either pour at the point of the contact or through fertilization tube. That is the pythium example albugo. This is gametangial contact or gametangial copulation is there. So another one is the spermatization. So here spermatization means some fungi that produce many minute spore-like single celled structures are there. That is the spermatia. Non-motile gametes is called as a spermatia. Single cell spore-like structures, uh, these non-motile spores. So these structures are transferred from some vectors or means transfer agencies like water, wind, and insects. There are the different vectors also there. So these contain the migrate into receptive structure. These dikaryotic condition is established. Fucsinia. So Fucsinia, we can see these fermentation. So another method is the somatogamy. So this takes place so here in the fungi formation, the gametes is absent, but the fungi uh, astromos takes place between the hyphae and there is somatic cells to fuse to form the dikaryotic cell. See here that the diagram is also there. Okay. So these are the some general characters of the fungi. Okay. What is the reproduction and nutrition, ultrastructure of fungi, cell wall. Okay. Just few minutes, we will come back. So very thank you for waiting the class. So nice. So coming to the classification here. So here mainly so classification is means phylogenetic consideration is more important. Means the division of mycota or either fungi and molds are including the true slime molds. That is myxomycetes. And lower fungi, that is the phycomycetes, the higher fungi is the eumycetes. Means here the slime modes means rhizopus. These are the myxomycetes. So fungi be classified according to various parameters, include here the taxonomic hierarchy and classification based on the spore production and that is the classification medically important fungi and based on the route of the acquisition based on the valence. So here, the different hierarchies are there based on the here gymnomycota, masticomycota, and amasticomycota. This is also there. And here, Alexopolus. According to the Alexopolus and means they are proposed the fungal classification in 1979. Here, there are the placed in the different kingdom of Caryota means gymnomycota, masticomycota, and amasticomycota. Again, there are the divisions and subdivisions also, classes and orders also there. That is the first one is the gymnomycota, means it includes the phagotrophic organism, and this division is comprised of two subdivisions are there. That is the Acrasio gymnomycota and that is the Plasmodio gymnomycota, mycotina. These classes are there. It- Okay. Okay. This one is not sure. Some problem is there. Problem is after what happened for the wait, wait. Hmm. Okay. Hmm. So here is audible clear? Oh, okay. Can I continue?
Okay, sir. Okay.
Okay. Thank you. So some sharing issues are occur. Wait. Okay. Let's see, wait. So means that is the taxonomy of the fungi means based on the phylogeny means according to morphology, anatomical characters, nutrition, mode of nutrition, physiology, and chemical composition also there, and cell wall composition and molecular method. So here these are the based on these characters here we can consider in the there are different classes are there. So morphology means there is the color of the growth and the growth rate and mode of the spore production. These are the we are to consider and anatomical characters also. So anatomical characters here that is the internal detail of the fungi is observed. That is means what here spore producing structure and arrangement of assay are basidia and also sterile structure like paraphysis and hymenium. This is also there, one of the considered here. So here, based on these conditions, right? So these aspect here, that is the mycota means the mycota. There are the two divisions are means the sexual reproduction is not identified. Sexual reproduction are identified. Two categories are there. Mycota and divided into two groups are there. Means sexual reproduction identified. Another one is the sexual reproduction is not identified. So sexual reproduction is not identified fungi we call as imperfect fungi. That one class we call as the Deuteromycotina, Deuteromycetes, Deuteromycetes or Deuteromycota. So here another category is the sexual reproduction is identified, that is the Oomycota and oh, that is the means primitive fungi, and another one is the advanced fungi. Oomycetes, that one we call as Oomycetes or either Oomycota. So here there is the phycomestis, zygomestis are there, and algae fungi here conjugate to fungi, that is the Phytophthora albugo that we call as a fungal algal fungi, and Mucorrhizopus conjugate to fungi. So Eumycota, again here two classes are there, Ascomestis and another one is the Basidiomystis. Ascomestis we call as a sac fungi. Ascus are present. That yeast is there, Ascomestis, example yeast, and Basidiomycota or Basidiomystis, here club fungi or Fuchsinia agaricus. These are the Basidiomystis fungi. Okay. So here another um, classification here. So Answer's classification. According to Answer's classification here, this fungi here Myxomycota and another one is the Eumycota. Alexopoulos classification is there and Answer's classification is there. Two types of the classifications are there. Right? Is it okay? Audible? Clear? Is okay? Is anybody?
Okay, sir, audible.
Oh, okay, nice. So here, Alexopoulos classification is there, Answer's classification is there, two types of the classification. So Answer's classification here, that is the kingdom of fungi, Mycota. Mycota, they are divided into two divisions are there. That is the what here, Myxomycota and another one is the Eumycota. So here, Myxomycota means based on the plasmodium, presence or absent. Okay, Nadi. So the presence of plasmodium is absent or present. Based on that, here Eumycota and Myxomycota, two divisions are there. And Myxomycota again divided into four classes are there. That is the Acrasiomystis, Hydromyxomystis, Myxomystis, and Plasmodiophoromystis. And Eumycota here divided into another five subdivisions are there. That is the Masticomycotina and Zygomycotina, Ascomycotina, Basidiomycotina, Deuteromycotina. So Deuteromycotina we can call as the imperfect fungi. So Masticomycotina they are produced the juice spores, and Zygomycotina they are to produce the sexual juice spores. Here also Ascomycotina also sexual ascospores are produced. Basidiomycotina, that is the sexual spores, basidiospores are there. But here, Deuteromycotina, there is no sexual reproduction. Means that's why we call it the imperfect fungi. Lack of the sexual reproduction is in the Deuteromycotina. So, right? So, that today class we are to discuss general character and the classification of fungi. Is okay clear? Is there anybody?
Clear, sir.
Right? So then
Sir, can
another
you share
class.
the notes? Can you
Sorry.
share the notes? Can you please share the notes in the group?
Okay.
Slides, whatever you have to. If
So
possible.
group. How can I share in this in
Kumar
the
sir, you can send to Kumar sir.
okay. Okay. I will send to Kumar sir. The Kumar sir send in the group.
Sir.
Right.
Okay, sir. Thank you.
So nice presence in this class. Attention is very nice. And approximately around 30 people. Okay, 28 people. Right. So very nice presence to continue these presence only until the program. Right. Okay.
Thank you, sir.
Thank you. Nice. Thank you, everybody. Thank you so much. Right.